Good evening, everybody. Good morning, good afternoon, depending when you're watching. I should note that I do take that catchphrase from our friends at Apicultura Sin Fronteras. I often say that, so I wanted to acknowledge them. But welcome to tonight's webinar. Tonight's webinar is going to focus on double-stranded RNA for Varroa control. So we're going to have a number of guests. We have Devan Rawn from the University of Florida, Joana Merk from Victoria University in New Zealand, myself from the Manitoba Tech Transfer Program, and we have two representatives with GreenLight Biosciences, Adam and Brian, here with us tonight. So just to get started, I just wanted to mention this webinar is not going to go over too much of the basics on RNAi for Varroa control, but just to bring everyone up to speed on the same page, we're talking about a new active ingredient to control Varroa. GreenLight Biosciences produces a product called Norroa with the active ingredient called Vadiscana. It's fed in a sugar syrup, and this mite control treatment does not kill mites. What it does is it prevents mites from reproducing. And we're going to hear a little bit about this from our first presenter tonight. But before we jump into that, I just wanted to mention that if you are looking for some primers on Norroa and how it works, you could check out the North Central IPM Center's recent webinar on RNAi. You could also check out a recent webinar with Adam from GreenLight that he did with Garrett Slater from Texas A&M University. So tonight we'll be taking some questions at the end. Feel free to put them in the chat. I think it's best if you put your questions in at the end of each presentation or at the end of all the presentations, so I'll be able to find them more easily and ask them to our presenters. So in a moment, I'm just going to bring up on the screen Devan. Devan is joining us from Florida tonight. He joined the Honeybee Research and Extension Lab at the University of Florida in 2022. And now serves as the lead field technician. His work focuses on research in honeybee nutrition, control, and Varroa queen rearing. Devan comes from Ontario, so he received his Bachelor of Science in Biology from the University of Guelph. So we're really happy to have him here on this Manitoba-run webinar tonight. He is previously a technician with the Ontario Beekeepers Association, the Tech Transfer Program, and a commercial beekeeper producing queens, honey, and is taught at Niagara College in Canada's commercial beekeeping program. So we're going to welcome Devan here tonight. And Devan, I'm just going to pull up your presentation as soon as you click share.
Okay. Thank you. Okay. Great. Thanks so much for the introduction, Matthew. And yeah, thanks for inviting me and everyone who's tuning in. It's nice to be speaking to an audience that maybe mostly Canadian. I've been down here in Florida now for almost 4 years completely, and I love it. But of course, there's some things that I miss about home. So tonight, I really just want to walk you through the field trials that we did with this product. Before it was registered for use, at least here in the US, and what sort of results that we saw. So I'm going to dive right into it. Let me click here. Okay. So as a real brief introduction, should we still be worried about Varroa destructor? And of course, all of you that are beekeepers out there know that yes, we should be. Still the largest contributor to honeybee losses globally, and doing a lot of bad things in our colony, contributing to the spread of viruses, and in a lot of different ways making our bees sick. So that's where Norroa, this new product, comes into play. And so without diving into all the details about the RNAi process, I did just want to share with you guys a little bit about specifically what's being targeted in our mites. And yeah, to give you an idea of like what's the real mode of action happening here. So Vadiscana, the active ingredient in Norroa, is targeting a specific gene in Varroa destructor called calmodulin-like. And the sequence of the active ingredient is unique to that sequence in Varroa mites. So we're not going to have off-target effects on our bees or other things that may be exposed to this active ingredient like us too. So calmodulin-like, in this case, it's really responsible for embryogenesis and a lot of that developmental activity that's happening when the female Varroa is trying to create that egg inside of her and lay it inside the pupal cell. So I wanted to show you some of these pictures here. This is a living mite in this case who is getting prepared to lay an egg. And you can see how swollen these mites get, how the different segments of their bodies actually start to separate. That's because the offspring, in this case, the egg that she's about to lay, is a significant portion of her body weight at this point. Like something like close to half the mite's total body weight is going to be that egg that she lays there. And imagine this mite doing that, every 24 to 30 hours inside a cell for those days that the cell is capped too. It's a really impressive feat, and you can imagine it takes a lot of effort and a lot of things going just right. And so disrupting this calmodulin gene so the protein isn't translated is how we're targeting these mites. So that's the mite swollen. There's another view of a mite. They really do have to swell their bodies and separate these segments. And then this last picture, while not, I know, particularly not great photo quality, this is a mite that had actually died while trying to deposit her first egg. And this was a mite that was treated with the active ingredient. So along with field trials, we've been doing a lot of lab trials with the ingredient and raising mites in the lab. And you can see in this case, that mite is not swollen there. Her body segments are all back together. And this is all that she was able to come up with for an egg. And so it's definitely not nearly the mass that those other photos were showing the mite was developing inside. And we think this is a good example of a mite that was unable to successfully lay her egg because of the active ingredient there. Okay, so what did we do here in Florida with Norroa? I'm going to talk specifically about a pair of field trials that we tried to frame around whether there was a good honey flow coming into our colonies or no honey, no natural nectar coming into our colonies. Here in Florida, we keep bees year round, really. There's some let up through late November through mid-January, but in general, beekeepers in Florida are active 12 months of the year. And there are things that we can do with our colonies and get into them whenever we want to. So we just really work around different honey flows. Whether you're moving your bees for that purpose or they're in one spot, honey flows will be up and down throughout the year. So that's how we targeted the seasonality of these two trials. And then I'm going to talk to you about some very specific reproduction trials, looking inside the pupal cells and getting an idea of what sort of reproduction was happening with our mites. Okay, this is the paper that was published March or April or so this year. And so, sorry about that. This QR code will take you to the website where it's open access. It's free for everyone to read, which we're happy that we had the funding to be able to do that, make everything accessible. I'll show you this again at the end if you're interested in seeing all the details associated with our studies. Okay, each of these field trials used 36 colonies. There was 18 treated, 18 control. We did frame it also with these different sizes of colonies. So we tested nuke, singles, and double brood chamber colonies. I'm not going to, because of time, really get into the results of the differences between all of those treatment units, mostly because in general, Norroa worked very similarly across the different sizes of colonies that we used. So I'll really just keep it between that nectar dearth and the nectar flow trials from here. But you can read the full paper to see any small differences that we did tease apart in that data. Okay, this is the product on top of hives. We applied it, in our case, on the top bars. I know the label now says that it can be put under colonies on the bottom board, and bees can feed from the pouches that way too. In our case, we put it on the top bars with a spacer rim before our lids on all of our colonies here now. Okay. So one of the terms that you might hear quite a bit about when dealing with RNAi technology is the term gene knockdown. When we say gene knockdown, we're really talking about from the point where the DNA in the nucleus of the cell is being read before it gets translated. The RNA moves outside of the nucleus and gets translated into a protein. We're talking about disrupting that process right in the middle. And so that's what we're kind of referring to as gene knockdown. How effective are these mites still able to make this protein? So in our field colonies that were fed the treatment, we were out there digging into the brood before the treatment on day 3, day 7, day 14, and collecting mites out of the brood food. So I'm targeting these like L5, the oldest possible larvae that are in our colonies, and pulling that larva aside, digging out the brood food from there, and sampling the mites that are swimming in that brood food, which we can assume and hope is diluted with some of the Norroa product. And that's where we're really exposing our mites. And so we relied on help from the people at GreenLight Biosciences their scientists and lab technicians to get this genetic work done. Okay, this is just a fun picture, I think, because in our second field trial, we dyed the product. And so we really wanted to see where was the product going through the hive, how quickly were the bees taking it up and using it in the brood food, and what did that sort of look like as a concentration. So we used the dye sort of as a proxy for the concentration of the active ingredient, and I'll show you some results on that towards the end. But you can see these bees feeding on the pouches normally, and the red tint that they get in their abdomens from filling up on that dyed sugar syrup. So I just like that picture. The final reproductive trial was more specifically looking at, are these mites reproducing? Do they have offspring at the proper stages where we expect them to, based on the timeline of the pupa that's in the cell that we're uncapping? So I'll get to that. I think this, even more so than our the sort of larger overview of the field trials, this really showed how the product was working and disrupting mite reproduction. So yeah, I'll show you those results at the end here too. These charts are, really boil down the crux of our field trials. The important thing to notice, we've got a nectar dearth trial on the left and the trial during a good honey flow on the right. But noticing where we started our mite infestation levels, I think, is the more important part of our results here. During the nectar dearth trial, we started on average about 4.5 mites per 100 bees. And throughout this trial, 3 months later, even our treated colonies continued to grow. They did grow at a slightly slower rate than the control colonies, but there was probably just at a level where there were so many mites in these colonies to begin with that some of those mites avoided the initial treatment and were relatively unaffected, and so they still continued to grow throughout the trial. Where we really saw the best effect of the product was when we started at or slightly below the treatment threshold of 3 mites per 100 bees. And during that trial, 3 months later, 12 weeks that we sampled across, our treated colonies still remained at the same level of mites that they started with. They did actually, during the first 3 weeks, decrease in mite levels and really diverged from the control colonies there. But even after 12 weeks, they had only rebounded to the point where they were originally. So the product was really able to maintain low mite levels when the mite levels were relatively low to begin with. It is a bit odd here. In our control colonies, we didn't see much growth in these last 3 weeks of the trial there. I might have expected those control colonies to really just ramp up at that point. So I can't exactly explain why this is a flat line there, but these are still, we saw significant differences between our treated mite levels and our control mite levels there. Okay, this is the gene knockdown data. And so, like I said, this is talking about how well these mites are able to translate the gene for this protein into the actual protein itself. So we measure these sort of normalized to the control mites. So the dark blue bars are the treated mites, and the light blue bars is the relative expression of the control mites. You can see in our first trial here, we saw 1 week after the pouch was applied, 7 days later, we had a 60% reduction in how much of that gene was actually making it to protein translation. Yeah, so they're only doing about 40% as much as the untreated mites. Our second trial, when the nectar flow was on, despite this one showing the more effective mite control overall, we didn't see quite as strong of a gene knockdown. There's a couple things to explain that. I mean, we framed this whole study around the nectar flow because there's some suspicion that if this active ingredient is delivered in a sugar syrup, then natural nectar coming into the hive is just going to serve to dilute that active ingredient. And the brood food, depending on the age of the larva, what they're feeding, it can be more than half nectar or honey that the bees had previously stored if there's no nectar flow. But when the nectar flow is really on strong, they'll often just use that raw nectar diluted with their royal jelly and fed to workers that way. So that does kind of explain when a lot of nectar is coming into the hive, it may be diluting the product a little bit, but we saw actually the better field efficacy in this season anyway. So I don't think it's anything to be concerned about. And this picture I think shows it's an interesting case with this product. We dyed it, and we saw the dyed product in our brood food, which I'll show you the charts of in a second. But we also saw the bees storing it, and so it's still a question, like, is this active ingredient, how long is it going to be effective for our bees if they're storing it on the outsides of the brood chamber, on your outside frames, and using it months down the line? I'm not sure about that, to be honest. Okay, and this is the dye concentration in our brood food. So as I pull off those really old larvae, there's often, or always, I guess I should say, in a healthy colony, there's always residual brood food left under those larvae. And we can see how pink it got in our study. So interestingly, that really peaked at day 3. 3 days after we fed the pouch, it was the most red in that larval brood food, and then it tapered off. But even 2 weeks later, there was amounts of pink that we could see in our brood food. So that was really interesting to see. And then this is a real in-depth look at how well our mites are reproducing. One of the good things I think about working with Varroa mites in the lab is, while they're difficult to work with, at least they should be predictable. So the female mite lays eggs on a very specific schedule, like down to the hour plus or minus. And so based on the age of the honeybee pupa that we're uncapping, we should know exactly what sort of mite family is in that cell. And so I really think here this last column where we saw one male and a female nymph, this was the column where we knew that mite would be on track to reproduce successfully and have an offspring, a female offspring that would be mated and go on to reproduce herself in the hive. And so this is where we saw some of the biggest differences between our treated mites and our untreated mites. Naturally, our untreated mites were successfully at that point 80% of the time, whereas our mites that were from the colonies that had been treated with Norroa were only at that point 36% of the time. So a little more than half reduction in the successful mite reproduction. And this isn't direct application to mites or some kind of contrived lab study. These are, again, colonies that were just fed the product like every beekeeper is going to do, and then we were picking them out of the cells. Okay, so I thought that's a good representation of how this was really working. So you can go scan that QR code again if you want to go directly to the paper that all this information is from. And yeah, the last note about how, this is really, I just think, a good thing in general that we have another tool open for us. When you use it and how often you use it or how you use it is obviously up to you as beekeepers, but we can't complain about having another option available to us. So thank you very much. Hopefully I didn't go too far over time there. I was trying to get through it. This is at least a somewhat recent picture of our lab. You might notice Dr. Ellis there on the left. He is north of the border, not the national border, but he's north of the Florida border up in Georgia now. So it's a little bit different around here, but things are still going strong. We got a lot of people here at the lab. So thanks again, Matthew, and everyone for watching.
Thanks, Devan. Yeah, thank you very much. And if anybody's got questions for Devan, you can put them in the chat now or hold them to the end, and we'll come back to everybody at the end of all the presentations. So next up, I will invite Joana to the screen. Joana Merk is a PhD student based in Wellington, New Zealand. Her background is in veterinary microbiology and has a degree in veterinary medicine from Munich, Germany. She started her PhD at Victoria University in 2023 and is a part of Dr. Phil Lester's research group, who works in entomology and pest control. So today she's here to talk to us about her study looking at RNAi-based control of mites and its impacts on honeybees. So I'll just add your presentation up on the screen, and there you go. You can scroll back maybe, and then.
Yeah.
Perfect.
All right.
Here we go. Okay. I'll leave you to it.
Thank you. Thank you, Matthew, for introducing me, and thank you for having me. Yeah, as you can tell from my title, I'd like to tell you a little bit more about Norroa tonight and its effects on individual honeybees and their health. As you can see also, I'm only virtually here with you. I'm actually based in Wellington, New Zealand. So I thought I'd start with a little overview over our situation with Varroa, because it might be a little bit different from other countries, wherever you're listening from. In New Zealand, Varroa was detected quite recently. So our first incursion was in 2000, quite a bit more recent than other countries. And here, Varroa are responsible for about 50% of our colony losses. As you can see from the graphic to the left, most of our reasons for colony losses stay constant over the years. So we have the usual suspects: queen problems, starvation, wasps. But we see that hives lost to Varroa are increasing over the last few years, as you can see from the rising line. So we're wondering what's going on with our New Zealand hives. And one of the most likely causes could be that we have resistance in our mite populations. So maybe our mites are getting resistant to conventional treatments. So we asked beekeepers, if you lost a hive to Varroa, what do you think was the reason? And most beekeepers still say they probably treated at the wrong time. So if you look at these sectors, green and purple are beekeepers saying, oh, I treated at the wrong time, or mites reinvaded my hive. But about 1/3 of our losses, and that's the red and yellow sectors, beekeepers say my product wasn't effective or my dosage was too low. So we have some evidence here that New Zealand mites are getting resistant to our conventional treatments. Probably still a lot less than in the US or other countries, but there is something going on. And that's why also we in New Zealand are looking for new products. And again, that's when Norroa comes in. Everyone else has already done the heavy lifting for me, so I'm going to keep this quite brief. We also did a couple of trials with Norroa in the field, but also in the lab. We saw the same effects. It targets the calmodulin-like gene and it inhibits reproduction. The application method is obviously these pouches that you put on top of your brood box, and that is relevant to us because that delivery method means that Vatiscana is being delivered to the mites by the bees, right? We have nurse bees that actually bring the larval food into the brood cells, and to expose Varroa mites, the larvae are also going to be exposed. The good news is it seems to not affect larvae whatsoever. So if you look at the bar graph to the right, here we look at larval survival after the hives were treated with Vatiscana. So blue and green bars show Vatiscana treated hives, and orange and yellow bars show untreated control hives. Great news, there is no difference in survival for larvae, so it seems like they are not affected by Vatiscana. It's really specific to the mites. But this brought us to our next trial. So we also wanted to investigate if there are any effects of the product on adult bees after they hatch. And so we came up with this trial. For this trial, we wanted to look at the lifespan of adult bees to see how long they're surviving in their hive. We also wanted to look at the foraging behavior, so to see if there's any behavioral changes. And we also looked a little bit at mite populations in our treated hives. This was just a minor thing. We only did bottom board counts. So I just have to say that right here. For my setup, as I said, we're based in Wellington. So we use 2 apiary sites around the Wellington campus. And I used 3 different treatment groups for this trial, each with 3 replicates. So we had a control group that received no mite control whatsoever. We just fed them blank sugar water pouches. Then we had a group that was treated with Amitraz strips, just commercial strips that you've all seen and used before. And the 3rd group actually received those pouches containing Norroa with the active ingredient Vatiscana. So I wanted to track bees from all these hives, and I did that using RFID, or radio frequency identification technology. It sounds complicated, but really you just have a tiny RFID chip, as you can see it in the picture, attached to some of your bees. And then to each of your hives, you have one of these white boxes, an RFID reader attached to the hive entrance. So whenever one of your bees enters the hive or leaves the hive, this reader will detect it. To kind of get maximum exposure, we treated the hives first with Vatiscana or the other treatment, and then we collected newly hatching bees. So we collected them directly from the frames when they came out of their brood cell. Then we tagged them with these tiny RFID chips. So we aimed for 150 bees per hive. And we assigned them to the home hive, so we would know which hive they were born in, hatched in. And then we just released the tagged bees back into their hives just to live their full and happy life. And I brought a little video here just to show you our system in action. So the white box you can see is the RFID reader. And then you will soon see 2 bees leaving the entrance with their tiny chips on their back. So this is number 1. Yeah, right here. They're behaving completely normally, so it seemed like the RFID chips are not disturbing them whatsoever. And we can also see the other bees interact with them normally. So we'll soon have one returning to the hive. They fly just fine. The RFID chips are really light, less than 5 milligram, and they can fly with them. And also the bees, their nestmates, don't recognize them as foreign or anything. So it seemed to be a really less invasive technology, but super accurate. You get all these counts. You can track all your foraging trips. So actually really neat. And with this, I can show you some results. So as I said, we first looked at lifespan. You can see the lifespan here on the horizontal axis in days. And then on the vertical axis, you see the proportion of bees that is alive at each of these time points. So we found the shortest lifespan here in yellow was the untreated bees. So yeah, parasitization with Varroa really had negative effects on the bees, made them die earlier. We found that Bordetella-treated bees in green were intermediate, and Amethyst-treated bees, the red line here, actually survived for the longest. So they had an average lifespan of about 31 days. Again, my data is just a very quick overview. If you'd like to read the full paper, there is a DOI here included. If you just type that in any search engine, it'll come up with the paper, and you can spend more time looking at the graphs if you'd like to. So yeah, intermediate lifespan. I do think I have an explanation for that, but I will have to show you a couple more slides first, so bear with me. Next, as I said, we looked at foraging behavior. I looked at the average number of foraging trips that a bee did in her lifespan. So on the vertical axis here, we're looking at the number of foraging trips. We can see, and this was actually significant, that Bordetella-treated bees in green did more foraging trips than any other bees, so more than the Amethyst-treated and also more than the untreated. Pretty good news. So I looked at the average length of these foraging trips. So how long does it take a bee to come back to the hive? And here we're looking at the trip length in hours. And Bordetella-treated bees actually had the shortest trip lengths. If that doesn't impress you right now, let's look at the untreated ones. So untreated bees had the longest trip lengths. But as you can maybe see from the top box, all these little black dots are foraging trips that took them over 12 hours. Staying out over 12 hours from your hive is obviously not normal and means that these bees didn't return to their hives overnight. So very abnormal behavior, and probably means either the bees were too weak to fly back at night, or maybe they got disorientated and couldn't find their way back to their hive. Either way, this is not normal behavior and probably not good. So the Bordetella-treated bees, on the other hand, were very quick with their foraging trips and pretty much always returned back to the hives is actually a good sign. So altogether, what we can see from this foraging behavior analysis is that Bordetella-treated bees were actually the most active foragers and did the most trips. And I think that can actually also explain our shorter lifespans, because obviously foraging behavior is very risky. It forces the bees to leave the safe hive. So if you're more out in the open foraging, some of them might get killed, might have a shorter lifespan and don't return back to the hive. So yeah, I think if you're a very active forager, that probably correlates with a slightly shorter lifespan. So here we got some mite counts. As I said, we only did bottom board counts, so we didn't have to open up the hives and disturb the tagged bees. Not much happened for the first 8 to 10 weeks, and then mite loads started to increase in all hives. That's mostly due to the season, because we were just going into autumn. But we found significantly lower mite counts in the Vatiskana treated hives. So we can see that we really had a positive effect of Vatiskana on mite loads here. And just a little side fact to the end. So I tagged all my bees so I would know which hive they belonged to, but I also found that my bees were not only going back to their own hive, they also went around and robbed other hives. And actually I detected some very naughty bees robbing not only their immediate neighbors in the same apiary, but they also went out and robbed the other apiary with their more distant neighbors. And if you look at the little graphic to the right, I found that I mainly had one really naughty hive, which was hive number rate 3, and they just went out and robbed all other hives. They started a really a robbing frenzy at university. So I found that's just a cool result. That's just cool to see how different hives are and how RFID tracking can show us what our bees do all day. So yeah, that brings me to the end of my talk, and I just want to leave you with a couple takeaways from my study. We found that Varroa control, both Emithras, Vatiskana, whatever's working for you, also improves the health and the life of our bees. They live longer. We found that Vatiskana treated bees were actually more active as forager. They did more foraging trips, so that's good news. Vatiskana seems to not affect or actually positively affect foraging behavior. And as I said before, foraging is probably associated with various risks. So you might get an active foraging behavior at the trade-off of a slightly lower lifespan. And with this, I just want to say thank you to my lab group, both Mael and Rose, who helped me tagging all these 1,500 bees within a week. My supervisor, Phil, who did all the camera work, all the videos you've seen. Of course, GreenLight Biosciences for the product, and Matthew for organizing this, and any and everyone who's listening. Thank you all.
Thank you so much, Joana. That was excellent. Thank you.
Thank you.
I will just continue now with the final presentation of the night, which is going to be myself presenting on Norroa field trials here in Canada. So just want to make a disclaimer. Although GreenLight Biosciences has registered Norroa in the United States of America, it is not registered in Canada at the present time. So this product cannot be manufactured, imported, distributed, or used in Canada at this time, unless explicit authorization has been obtained from Health Canada to use the product for the purpose of conducting research under the Pest Control Products Regulations. So GreenLight has been supporting trials of this RNAi Norroa product here across Canada since 2023. And there's still 3 trials ongoing this year in 2026. It's been done in 3 regions, Alberta, Ontario, and Manitoba, for a total of 10 trials and using over 700 colonies. So in Alberta, these trials have been led by Lene Oving with her consulting company and with a participating beekeeper. In Ontario, these trials were led by the Ontario Tech Transfer Program, with Melanie Kempers, and later with Brittany Kyle. And in Manitoba, these trials were led by our tech transfer program, by myself. So I'm going to tell you a little bit about our results here in Manitoba based on 2 years of experiments. So it's going to be a Manitoba focus. I'm going to start with 2024. So in 2024, we used 64 colonies. We set them up on May 17th, and the first treatment was applied on June 25th. The trial ended October 11th, and the purpose was to evaluate the efficacy of Norroa for controlling Varroa. To set up my colonies, that spring I had about 100 colonies of different strengths and different mite loads. So in order to standardize, equalize all the populations, what we ended up doing was catching all of the queens, figuring out all the mite counts, and then we shook all of the bees, all the hives, into 2 separate boxes. One box had the high mite load count bees, and the other box had the low mite count bees. This way, we were able to then make up packages by equalizing the bees and the mite population, so we could have a standardized mite pressure across all the colonies. We added new queens, all of the same origin. So in 2024, we had 4 different treatment groups. We had Norroa as a standalone spring treatment on May 25th, Norroa plus oxalic acid dribble 5 days prior. And we used an industry standard, amatraz strips in this case, as well as an untreated control group. So there were 16 colonies per group for 64 colonies. We collected data on various assessments, which I'm going to take you through and show you what we found. But we collected mite washes. We collected bee samples to do Varroa infestation level estimates using alcohol washes. We counted frames of bees, frames of brood. We did open and closed brood scores, and we looked for any observable signs of disease, such as deformed wing virus, chalk brood, etc. So starting in 2024, the average mite load was at 0.6% across all groups at the first assessment date, on treatment date on June 25th. I think I may have said in the past slide we applied the treatment on May 25th. Actually, we set up the colonies May 25th or early May or mid-May, and then we applied the first treatment and did the first assessment June 25th. So at that point, we had 0.6% of an infestation level. There was no statistical difference in between the different groups, the starting point. By August 1st, at the second assessment, we saw the control group at 1%, the amatraz group at 0.5%, and the 2 Norroa groups at 0.3%. So at this point, mites are kind of doing what mites do. They're descending into the large number of brew frames in the middle of the summer, and there's likely a little bit of control happening there. So now at the next time point we're going to look at is when things start to get quite interesting as we move into September. So in September, we saw that since that June 25th treatment, the Norroa had held the mites down to 2.4% and 3.2%, the amatraz strips at 5.5%, and the untreated controls were up to 8.5%. So at this point, most people would think, well, your trial is over, your control's at 8.5%, your Norroa has controlled the mites to just around 3%, and you could take care of your colonies and kind of treat them and move on with your life. But we really wanted to put these products to the test to see how long the different treatments would hold the mites down compared to that untreated control. So this trial continued for another 4 weeks. I should just note that at this point in the trial on September 13th, these are obvious difference in mite levels to, the standard beekeeper looking at 8.5% versus 2.4 or 3.2%. But we did also see a statistical difference in those mite loads between the controls and the amatraz, as well as the controls and the Norroa groups. So on October 11th, the average for the untreated controls were up at nearly 30%. The amatraz strips had held the mites down to 19.4%, and the Norroa had held the mites down to 12.6% and 10.2%. So there was a statistical difference in between the control groups and the Norroa groups, as well as the amatraz groups and one of the Norroa groups. So again, the black there on the bottom is oxalic acid and Norroa together, and the green is just Norroa. And there wasn't any statistical difference in between those two groups. And from what I understand from GreenLight, they haven't really seen a difference whether you're treating with oxalic prior or just a day before, or in this case, Norroa only. So a couple of things I just want to note about this trial, because we definitely thought we saw the amitraz underperforming with only holding the mites to 20% by the end of the trial. We did do some field bioassays, so doing the cage bee testing to expose the bees with mites to a strip to see how many mites that strip would knock down. And we sent off mites that we collected for genotyping to look for mutations associated with resistance. And of course, we have the field data. And we saw about approximately a 50% level of resistance to amitraz in the mite population that year. That's going to be different every year in every mite population, and that can fluctuate on your treatments, but that's what it looked like this specific year here. So that's why we thought maybe that amitraz was underperforming. Now, moving on to the 2025 trial last year, we continued with using 64 colonies. We wanted to align it a little bit closer to a typical Manitoba timeline, so getting that treatment on a bit earlier. And we wanted to compare it with a different miticide. So we wanted to standardize those colonies again with reasonably elevated Varroa levels in order to test the efficacy of our mite control products. So we actually bought package bees and we installed them April 14th. And then we made sure that there were a sufficient amount of mites in those colonies and standardized pressure across all the groups. Our treatment did not begin when we started with the packages. It began once those packages got developed a bit and the colonies were on their way. There was plenty of open and closed brood in the colony. We want to put that Norroa in so it can come into contact with the open brood, reach the open brood. So that's why we need to wait a little bit longer instead of treating, for example, when it's broodless. So we began on May 22nd and we wrapped up on September 3rd. So I'll take you through the results of last year's trial. For the treatment groups, we used Norroa with 16 colonies, one treatment group, and we used Norroa on another treatment group with another 16 colonies. Now, the reason I've separated these in two here is because during this trial, we had different plans for one of the treatment groups later on in the year, but those results are not part of the tonight's presentation. So all you need to know is that there were two Norroa groups. We also had an industry standard. We used Formic, and in this case, we used Formic Pro, so a slow release Formic. And we used the one pad every 10-day application rather than the two pads for 14 days. And of course, we had our untreated control, which was just fed sugar syrup with nothing else added. So on May 22nd, we had our first assessment day and treatment. The starting average across all groups was 0.9%. The control group actually had quite a bit lower mites, but not a statistical difference there. And then on June 30th, when we had our next assessment, we saw that the mite levels actually went down. So again, presumably, a lot of those mites went into the brood nest, expanding brood nest, and presumably there was some mite control happening there. The control was at 0.4% on June 30th. The Norroa groups were at 0.25, and the Formic group was at 0.04%. So that 0.04%, I think, represents like 2 mites that I found out of 16 colonies, so quite low. Moving forward to the next assessment, we start to see a little bit of difference here. On August 13th, the untreated controls are now up at 2.1%. The Norroa groups are at 0.9 and 0.8%, so just below that 1% sort of threshold and well below the 3% threshold. And the Formic group was at 0.4%, so Formic's still holding the mites quite low. There is a statistical difference between the untreated control at this August 13th time point and all of the other groups. So moving on now to the September time point, our final time point of the year, we saw that on September 3rd, the mites in the untreated control were up at 8.4%, and Norroa had held the mites down to 2.9 and 2.6% respectively, just under the 3% sort of threshold. And the Formic Pro had held the mites very low, down at 1.1%. So I just want to mention that in terms of the other treatments, when we're looking at the impact of the treatments on colony growth, they were all the same, except so we saw a bit of difference with the formic group. Here on this chart, we have weeks after treatment on the bottom. So at 0, they're all the same in size, about 6 frames of bees each colony. At 6 weeks after treatment, we saw pretty standard colony growth, but the formic group did not grow as quickly. However, it did recover by the 11-week point, and all treatments were similar from 11 weeks forward. Now, I just want to mention that the reason we're probably seeing this is these colonies were treated at six frames of bees, and that is the absolute minimum that, according to the Formic Pro label, where you should be applying that treatment. So it likely set the colony back a little bit, and we did see a reduction in honey production on that first round of honey. We did not measure honey production in this trial. However, it's quite clear that we missed some honey there on that first round. The colonies recovered beautifully later on and made plenty of honey. And as you could see in our previous slide there, they held the mite levels quite low all year. So that's just the only thing to note about the various other treatments in that trial. Overall, the Canadian experience here with Norroa, what have we measured? We've looked at efficacy, specifically with those elevated mite levels, to put the pressure on to test those products. We've kept track of colony growth and the general health statuses through the trial. In Manitoba, we used singles, starting with 6 to 8 frames of bees at the treatment date, depending on what year, and used colonies, again, with elevated and equalized mite levels. In terms of a bit further on the Manitoba experience and sort of integration into our IPM systems and trade-offs with other miticides, for testing Norroa here, I'd love to be able to see it tested when we're working with optimally low mite levels to see the type of control that we're getting in the future. Different colony sizes. I know Devan mentioned they were doing some of that in Florida, so that's really exciting to see. But I'd like to see, in terms of my own first-hand experience, testing it on different colonies. Some of that was done in Alberta. Alberta did use doubles in some cases. Local nectar flow and feed timing and integration into that. Actually, since I probably made this presentation, we've gotten a lot more experience with that, and this year's trial is going to tell us a lot more about that. Again, I was very interested in Devan's presentation in terms of measuring that in terms of dearth versus nectar flow. So looking forward to hear more about that and see our results from this year's trial. Environmental conditions, just thinking about when we apply products like Norroa, a lot of other products do have these temperature constraints and different environmental conditions that might make it the best time to apply. When we're thinking about using Norroa, definitely it's a little bit of a different thought process. We're not looking for a knockdown with the mites. We're looking to prevent these mites from reproducing and hopefully not having negative impacts on our colonies and targeting colonies when those mite levels are low. So all in all, we've tested a few different treatments. We've run Norroa now for 3 years. We're on our 4th. As all beekeepers know, when we're considering mite control options, we're thinking about labor, time investment, the cost of doing these things and the cost of the product, and then that return on investment. So are we getting the honey crop we want out of it, or are we getting the populations we want out of it? How's the bee health? So just trying to tie all those things together and understand where Norroa fits into our IPM system. It's really exciting. So I'm going to leave it here, and we'll have a little bit of conversation with our presenters, as well as with GreenLight Biosciences, and we could talk maybe a little bit more about that. So if you have any questions, put them in the chat relatively quickly, and we will address them. So I'll bring everybody else back on the stage now. Oh, and I guess I should go back to my slide and just thank you to Sustainable Canadian Agricultural Partnership, GreenLight Biosciences, and all of our other partners, and my contact information. All right. So, folks, I will now redirect my attention to some of the questions. But first off, yeah, I was just really enjoying everyone's presentations. Fascinating in both fronts. Devan, with the nectar flow and kind of your interpretation that in that study, there wasn't as much maybe to be worried about as we may have thought with the dilution. At least that's what it looks like your study indicated to us. And just fascinating how the bees are storing it and the color you used in that. So I definitely have to look closer if that's involved, if that's included in your paper.
Yeah, it's there. Actually, I saw a question here that maybe came up during my presentation. I don't know, where someone asked, has it been approved for use during the nectar flow? And it hasn't. That's right, Brian? As in, like, I kind of wanted to make this distinction. If you have honey supers on and you intend on collecting honey that you're going to harvest, that's not the time to use this product. Am I right to say that, Brian? It's not intended for that use at all.
Yeah, we recommend that you not treat with the honey supers on, and it's not a safety issue in the honey, but we don't want the product moved up into the supers, as your paper rightly shows. We want it stored in the brood chamber, so
right.
We don't want the honey supers on at the time of treatment.
Yeah.
You put the supers on as soon as the pouches are empty and the bees have stored it. So.
And so the distinction I wanted to make was, I know obviously in a Canadian climate, when the honey flow is on, you're making honey. That's your intention as a beekeeper, and that's not the case everywhere. Where here in Florida, when we get these up and down honey flows, there are times of year where beekeepers will not have honey supers on, maybe managing their colonies in double brood chambers, getting them ready for winter with the intention of maybe splitting them down before almonds the following January or February. And they really do encourage the bees to use that natural nectar as just storage or build up or sustaining themselves. It's not honey that's intended to be harvested. And so that's why we wanted to test the product during those conditions here in Florida, because not every honey flow here is one that's going to be harvested honey. Sometimes it's just bad honey. We get this Brazilian pepper in the fall that I can't stand, and the beekeepers are happy to just let the bees have that in a lot of cases.
All right. And I guess I'll just stick with you for one more thing, Devan, and it might also be directed to GreenLight here. Had a question, I'll just bring it up on the screen. Can you speak on a commercial scale application of Norroa? Now that came up during your presentation, Devan. I don't know if you want to talk about anything like that, or GreenLight maybe could mention about what they're seeing and hearing at the commercial scale right now.
Well, yeah, my real quick impression anyway is, from a labor time cost point of view, it's just as easy to apply, user friendly, and doesn't take up a lot of your time or a lot of extra labor to go around and put this on top of your hives. Basically the same as putting in apivar strips. So from that point of view, it's, in my mind, better than some of the more labor-intensive treatments like oxalic acid vaporization. Can be a little more cumbersome. So from that point of view, I think it's a good thing for commercial beekeepers.
Yeah, and I can step in just on what we're hearing from commercial beekeepers in the U.S. So I live in North Dakota, and this past weekend we just had our state association meeting combined with Minnesota and South Dakota. So we had a lot of commercial beekeepers there. There's definitely people trying it out on a commercial scale. Obviously, with something brand new with no experience, no one's really eager to put their entire operation on something brand new that they have no experience with. But they're definitely trying it out. Early responses are very positive. Everyone's really happy with what they're seeing, with how it's working for them. I did have one beekeeper that's just about 45 minutes north of me realize that if things are going the way they appear, he can probably skip the knockdown that he normally has to do in the middle of summer. So So far, it's been going very good.
I have another question here that might lead into Joanna's presentation. Now, the person asking here just wanted to confirm that this product is designed for the suppression of reproduction in the mite. It's not a knockdown, which we can confirm. And they're asking about the viral load, and that just makes me think of Joanna's presentation with the untreated controls, the shorter lifespan, and that type of thing. Has anyone worked, or have you looked at results on the viral loads compared to the controls?
Not in my study specifically, but I know me and colleagues have done viral loads for untreated controls, and I think that definitely plays into bees being less healthy with higher Varroa loads. That with higher Varroa loads, you obviously also get higher deformed wing virus loads, and deformed wing virus is really probably one of the biggest drivers in this early death, shortened lifespan, the disorientation and learning disabilities or issues that these bees experience that I saw. So I didn't measure them. I can't confirm that, but other studies have definitely shown a link in between Varroa, deformed wing virus, and then disorientation, shorter lifespans, lower bee weights.
Excellent. Thank you. Yeah, I would love to look at RFID tags on bees all day. I mean, that's fascinating to see what they're doing, where they're going, and that robbing colony. Is there any more? It's kind of a side question here, but is there any more information that you've written up about things you've observed during that trial?
I only focused on robbing, but I also saw some drifting bees. So I actually realized there's a difference between robbing bees, so bees that exactly know that they're going into a hive that's not their home, and then they return back to their home, just stealing honey or nectar, and drifting bees, as I call them, that are like younger, confused bees on orientation flights. So usually they're around the time when they first go on the orientation flight, so somewhere between like 7 and 14 days. And they just get confused. They don't fly back into their home hive. They fly into a neighboring hive, and then they just live with them. They never return to their home. I found that quite interesting.
Yeah, I wonder if there would be is it possible in the future to look at if there's more of that happening with bees that have been under more stress, with an infestation and stuff like that? Or did you look at that in your study?
I didn't look at that in my study, but I would love to look at that. And to be honest, I really enjoyed the RFID trial. I would definitely want to do that again. And since we still have the equipment, maybe I can convince my supervisor.
Right. If any of the other presenters have questions for each other, go ahead. I'm just going to pull up now one question for myself just to address it. The question was, after I made the packages, did I let the colonies build up before I started the different treatments, or did I make the packages and put the treatments in right away? I made up the packages and then I let them build up again. So we have that regular functioning colony with open and closed brood, apply the treatment, and the treatment will be destined towards the brood to be able to control the mites.
Yeah, that raises an important point, Matthew. You need to have active brood present for this product to work well. It needs to get into the brood for the mites to be exposed. And so in that case, with the packages, starting new colonies and such, or after requeening, you want to let the queen get restarted, let her start laying before you treat.
Right. I'll make a final call out for questions here, and if there's not, we'll wrap up pretty soon. Does anyone else have anything they'd like to add that was maybe missing, or anything I think might be helpful to the audience? All right. Well, I'm looking forward to sort of rewatching these presentations when I can focus on not only running the webinar. I really appreciate you guys taking the time to be here and share this information with beekeepers. I get a lot of questions about Norroa. And I'm really happy to be here with GreenLight, sharing this work we've done together over the past years. And yeah, just putting it out there so beekeepers can see what we're up to and what's coming down the road, at least here in Canada. I know in the US this is already available, but not quite yet in Canada, and I believe not quite yet in New Zealand.
That's correct.
So unless you have anything else to add, I will thank everybody.
Thank you, Matthew.
Thank you.
Thank
Yeah,
you.
Matthew.
And for the night, thank you. Everyone can hear me?
Yes.
Yep.
Yeah.
Yes.
There we go. So it's